Chromatin is the most compact form for a undividing cell. In case of any undividing cell, chromatin is the highest packaging of the DNA. But in case of dividing cell, that means the cell that undergoes the division, either meiosis or mitosis, those kind of cell we have observed the further kinds of packaging and formation of chromosome. In this video, I'm going to discuss about the structure of chromatin, that means euchromatin and heterochromatin, what are they means for, and in the function of HAT and HDAC that allows the activity of the gene. Hello friends, welcome to Biology Concern. I'm Animesh. So let's jump into the topic. So first thing, chromatin is the ultimate compact structure for any kind of undividing cell, right? So if we closely observe a structure of chromatin, we found this kind of structure. So this kind of structure. Here you clearly observe that there is two different structure. One is more compact. So first and third structure are less compact. So first and third structure are less compact. Okay. And second structure, that is the second structure, is highly compact. Okay. So in this case, first and third structure are less compact and they known as euchromatin. Okay, they known as euchromatin. In case of second structure that is highly compact, they known as heterochromatin. That means in case of euchromatin, the nucleosome model, I already write down the structure of nucleosome. The nucleosome model are less compactly bound to each other, and here in case of euchromatin, we already observed here the presence of functional gene. Okay, functional genes are present in case of euchromatin, but in case of heterochromatin, there is no functional gene present. That means in case of heterochromatin, the packaging is highly compacted, and in this case, DNA is not allowed to separate from the nucleosome. That is why it is more compact and the structure is more compact, and it is known as heterochromatin. If we stain both of the part, euchromatin takes up the light stain. Okay, it is stained in light, but in case of heterochromatin, it's stained in dark, because here packaging is more. That means if packaging is more, that it consumes a stain more, or it take up the stain lot more than the euchromatin region. So this region are stained in dark, and this region euchromatin is region stained is light. So here those and these both regions are known as euchromatin, and this region is known as heterochromatin. Okay, and this is the centromere. This is the centromere. Okay, so this is the short arm, this is the long arm, heterochromatin and euchromatin. Okay, clear? Next thing, what is the function of HAT and HDAC? That means we already know that the presence of functional gene in the portion of euchromatin region, correct? So we have to activate the genes. That means gene is referred to as a segment of DNA that contains the information to produce a functional biological product, whether RNA or protein. So if a DNA portion can be transcribed into RNA and further translated into protein, those segment of particular DNA is known as genes. So how we activate the genes, or how cell activate the genes? Let's focus the nucleosome model here. Here you saw the clearly observed, here you clearly observe the spike are there. So those spikes are actually known as histone tails. Okay, so they are histone tails. All of the histones like H2A, H2B, H3 and H4, all of the histones come, all of the histones are provided with those histone tails. Okay, so first thing, HAT here comes into play. HAT stands for histone acetyltransferase. Okay, so HAT stands for histone acetyltransferase. It is an enzyme. HAT is actually as enzyme, and it works on those histone tails and acetyl, and add a acetyl, particular acetyl group in this particular histone tails. By adding those acetyl group in the particular histone tails, HAT actually convert the basic properties of the nucleosome or histone octamers. That means the basic properties of the histone octamers are, they are basic in nature. So they are basic in nature as well as they are positive in nature, positive in charge. So they are basic and positive charge. As I told you before, that histone proteins are consisted of lysine, either lysine or arginine amino acids. So lysine and arginine, those amino acids makes the histone proteins basic in nature as well as positive in nature. Okay, so positive in nature. Next thing, when a particular histone acetyltransferase add a particular acetyl group in each of the histone proteins, it actually shifts from base to acidic in nature. Okay, so basic to acidic in nature, as well as from positive to negative in nature. So the nature of nucleosome now has been changed, and as you all know, the DNA stands for deoxyribonucleic acid. So acid cannot interact with each other. Now the nucleosome has converted into acidic and also negative in charge properties, and DNA is also negative and also acidic in their properties. So DNA and nucleosome cannot interact with each other. So in this case here, let me give you a diagram on top view. So here, if we consider this one is H2A and this was in H2B, and this was in H3 and this was in H4. So in case here, the DNA is separated from the histone. Okay, so DNA is now separated from the histone core, that is the core histone or histone octamer. In this case, RNA is capable to bound with the particular DNA. So this is the RNA pol. So RNA polymerase is now available to bind, because here DNA is available to bound with the RNA pol. So transcription factors and RNA pol now bound with particular DNA segments, and they transcribed into a mRNA, produces mRNA and further protein. Okay, further protein, by which those proteins are come into play, and we considered this segment of DNA as gene, and this is how the histone acetyltransferase activates the portion of gene. After activating or after translation and after transcribing and translation of the particular gene, now the time when nucleosome have to shift back its original shape, okay, its original properties. So now another enzyme, HDAC, that means histone deacetylase, comes into play. So histone deacetylase now comes into play. When histone acetylase add a particular acetyl group in histone tails, then after completing its work, histone deacetylase removing those acetyl groups from the histone tails. Okay, so Histone deacetylase now removes those extra acetyl group from histone tails, and the nucleosome or core nucleosome that is consisted of H2A, H2B, H3 and H4 shifts back to normal position, and it again in basic in nature and positive in charge. So the nature of nucleosome shifts back original to, that means the basic in nature and positive in charge. That means now DNA bound tightly with that particular nucleosome. Okay, so this is the work of HAT and this is the work of HDAC. So histone acetyltransferase loosen the grip of the DNA from nucleosome, and HDAC that removes the acetyl group from the histone tails, and it helps the to formation the nucleosome again its original shape, and that is why, and that is how it forms a particular chromatin structure. I think this video is really helpful to understand you the basic properties of HAT and HDAC and how they are activating the genes. Okay, the activity of HAT, further activation of the genes, and this is the all about euchromatin, heterochromatin, and how functional genes are only present in euchromatin region. So if you like the video, please hit the like button and don't forget to subscribe. Thanks for watching.